Okay, so today we're going to talk about data fitting and errors and how we can confuse ourselves, really. So we'll start with this quote from Richard Henderson saying, you must underestimate how many new ways we can deceive ourselves. Here's a classic case in point. This is an fMRI of an Atlantic salmon, so people use this for functional studies of the brain. And what they did was they showed pictures of human emotion to this salmon, and they recorded activity in the brain. And you can see here that there was spots of activity. This looks all very, very interesting. There's only one problem: the salmon was dead. So this is really kind of showing you in this kind of field, the real problem was in the data processing, and actually it could give rise to lots of false negatives. So what we hope to do is that we're going to have some lovely data, and we're going to have some lovely mathematical models. We're going to put the two together, and they're going to fit very, very nicely, like here. And your prints will come one day of kind of publications. So first of all, we kind of need to think. We've got some kind of biological model, and we've got some kind of method. We've got some controls. We're going to ask, is it reproducible? Is your targets valid? And that's going to give you a physical model of what's happening in the biology. At the minute, there's really a big issue with reproducibility. And so this study by Bayer kind of showed that 25% of published academics papers could only be reproduced. Amgen could only reproduce 11% of kind of landmark studies. And in the Cancer Reproducibility Project, they could only get 46% success rate. So why is that? Is there a lot of cheating going on, or is there, really just people are getting results because they're doing it in a specific way and they're not reporting that? So with all this, and journals really expect you to be very, very clear on what you do. They need also to deposit your raw data, and you need clarity on how reproducible your data is. So reasons you could kind of fail to reproduce something could be that you have the pressure to publish. Obviously, you're here, PhD students, you've got a pressure to get that PhD done. You might be a bit slipshod. You might kind of take a shortcut. And in the Swedish saying, sometimes a shortcut is the longest way around. There can also be cognitive bias, where cognitive, say, confirmation bias, where you know what the answer is, so you're going to fit your results to that. You might have selection bias. You kind of go, well, I did this experiment 5 times, but I got the right result in one of them, so that must be right. You have to kind of think about, well, why did the other 4 fail? Bandwagon, that can occur obviously in groups where, if you get a result that's actually different to what the current hypothesis is in the group, you're going against your boss. You don't want to rock the boat kind of thing. But also bandwagon can also come into how you do experiments, in the fact that, lots of labs may have that magic buffer that kind of works. They've done everything in, but that might not work for your project. Cluster illusion is really looking at the noise and kind of going, oh, well, that's the data that fits. I'll just get rid of all that other extraneous stuff. And then reporting bias is, of course, you want to please your PhD supervisor or your PI, and you kind of only talk about the results that went right, not the ones that went wrong. Often the point of reproducibility is per methodology. And if you've ever tried to then look at a paper and try and reproduce what someone has done in another lab, it's really scant detail. I mean, personally, one of the worst ones I ever had was I was trying to refold this protein. I got the construct from the lab. It didn't work. I tried lots and lots of different kinds, different kind of variations. Went back to the lab and said, can you give me a protocol? And they went, oh, no, the guy's gone. It's been a long time. And I said, well, I couldn't get this to work. And they said, oh, well, you only got it done once. Published the paper. So, that was a complete waste of my time. And obviously, there's an issue of lack of statistical power, particularly more in, say, in comparative studies. Right, so we have a physical model. We're going to ask, what the signal is, what's our raw data, what are controls, how are we going to correct it, what the data model is, and we're going to fit data. So what are the assumptions, in that? What are the, is your signal representative? How precise and accurate is your raw data? Are your controls good enough? Have you got the right data and time? You're going to have a data model. You're going to fit onto that data. What's the assumptions in that? How are you going to, what program are you going to use? What's your estimates? How are you going to estimate the quality of fit? And then hopefully after all that, you get some nice data that reports back to the physical model, which is then going to inform what's going on in biology. Right, so let's look at this first section. So do you have the right signal? Here's a case of something I did where I was looking at the binding of a nucleotide to a protein here, and we got a shift in the intrinsic protein, or we got a shift in intensity of the intrinsic protein fluorescence. Okay, so that looked good, and we could actually then titrate that in after correction for the inner filter effect, and we could fit that data. So then we could say that, oops, sorry, let's go back. Then we could say that, yeah, that this is representative here. And so hopefully we're just fitting, we're going to find out what this KD is here, this first bit. So we could take the corrected data and then put it into a data model. And the assumptions here are that it's a one-to-one binding, and that the amount of enzyme substrate complexes formed is going to be directly proportional to the fluorescence change. And also we expect that there's not going to be any change in the total amount of nucleotide, because we expect this catalysis rate to be 0. And in this case here, I'm using a non-hydrolyzable ATP analog, so hopefully that's true. But of course, that may not be so simple. And it could be that, well, what we're actually reporting here is not just a simple binding, but also conformational change, or the enzyme could be in 2 different states. So that then, then the signal changes, then what's that representative? So you always have to be careful about what your signal represents. Here's another case in point where things can go wrong. This was, again, this is a fluorescence polarization study where we've got a reporter binding onto kinase. And so that then when you increase the concentration of kinase, you get an increase in the polarization. And this was the molecule here. So there's a fluorescence molecule here. And then there's this analog of a nucleotide here, which should bind in the active site. But one problem was that when we just titrated in then the fluorescein itself, you got a very, very similar curve. So what's happening then is it's the kinase is then just binding onto the hydrophobic fluorescein. So one way of kind of getting around of that is in a binding reaction, then is to see if here you've got free kind of compounds then binding onto the same kinase. Can you displace things with a known positive binder? And in this case, you can kind of see that in green, yeah, this gets displaced, but the red ones only get partial displacement. Does this mean then that really, these molecules are not binding in the active site? So a good question is, what are your controls? So what we kind of aim for really in terms of accuracy and precision is, so you think if we got the true parameter here in cyan, here we are getting more accurate. And obviously here, when we've got better precision, we're getting very, very close to the true value. That this is going to be the best idea. Your data is going to be very, very representative of the true values. This tends to be more the case of what you get. And so you've got to scatter around the true value. So you just have to do more data points to average. In some cases, you get a very, very accurate, very precise reading. It's not very accurate. So here, in this case here, well, all the data points are here, but they're quite away from what the true thermodynamic value is. But in cases, sometimes that's fine for assays. We're only looking at differences such as differential scanning perimetry, where you're looking at a TM. It doesn't really matter. It's actually the real hydrodynamic TM. You're looking at shifts when compounds bind. So what are these kind of sources of noise and error? Well, you've got to look at the kind of the signal magnitude relative to noise. Have you made things up with accurate pipettes? Get them always calibrated every year. Things may just be stochastic. So these kind of random errors, they'll average out every time you kind of do the assay. And hopefully you've got good signal change? Have you accounted for the background buffer effects? Is your concentration accurate? Have you got good temperature control? And have you got, reached time for equilibrium? Temperature control is something that people really avoid. You've got a plate in the plate reader and you push it in. I remember a case where a PhD student was not getting reproducible results. And it's like, well, what's happening? She's doing same assay, same proteins, these were proteins she'd even stored. One was done in winter and one was done in summer. And of course, the instrument she was using wasn't actually controlled. And of course, in the building we were there wasn't air conditioning like here. And so the actual whole experiment was being run at about 10 degrees different temperature. So always it's very, very good to have positive and negative controls if you can. So is your assay robust? I mean, usually in academia, we're we're doing an assay maybe once or twice or 3 times. We're not really at the kind of scale in pharma. So, we never really think about these kind of things. But obviously, if you're doing a high throughput screen here, then this is very important. And to get an idea of how robust your assay is, then there's a measure called the C prime factor, which is really then looking at really it's the ratio of the standard deviations of the maximum and the min, then over the total signal, really. And so that I can see that we're up to above this 0.7 is good, and it's consistent across all the pits at the end here. So that then you can be confident that any kind of signal in the middle here is actually something that's actually real. And if you want to look further into this, then there's this nice paper here from Ingles, and describes more ideas of how you then report on how good your assay is in terms of signal window or coefficient of variation, etc. So, okay, can you repeat your experiment? Hopefully you've got valid targets. Hopefully you've got a good method and authentic reagents. And the key point is documentation. As you've got to document not only for yourself, but also for those students and postdocs coming after you. So make sure that everything is written down. So let's think about, is your reagents good? Classic quotes, don't waste thinking time on dirty enzymes. Here's a case I did, which was initially very exciting, where I was working on this chaperone HSP90 and one of its cofactors added in called FKBP59. And that seemed to really stimulate the ATPase activity of this human HSP90, which has got a very, very low rate. And so that looked really, really exciting. Whole ideas about what this could actually mean. Until then, I further purified the protein. Oh, let's go back. And you can tell this is old school chart paper. Okay, so here we go. Further purification. So this is the protein here, and this is done at further purification ion exchange. And you probably can see here, might be able to see there's 2 peaks here. And with the FKBP protein here, it's a big black blob. And then there's this fraction here, fraction 39, and you can't really see any protein there hardly at all. But then when you put it in the ATPase assay, here's HSP90, here's FKBP by itself, and then fraction 39 by itself, huge activity. So it was really contaminating ATPase in there. So that was that project was back to the drawing board. Okay, background signals, something we always, forget about usually, but it really can be very important. It could contribute to, say, like light scattering in fluorescence, or you could get heat of dilution in ITC. Here's a case of ITC here, where this is just DMSO. And that looks like, whoa, that looks like a nice curve. But really, it's just a mismatch between what was in the syringe and what was in the cell, just a slight mismatch, and you get a nice big heats of dilution here. In some cases, then, like in AUC, you need buffers to be exact, and similarly with SPR. And also try titrants can actually affect the signal. So if you're putting in an ATP, then that's going to absorb in the UV range. You have to be careful about that. Right, so data and time. We kind of always need to think about that. You know, have we got enough range, and have we done it for long enough? So here's an example from the literature of a high throughput screen using SPR, and you know, they fitted everything, and you know, is this real? I mean, you know, some of these you could easily just fit with a linear function. So in the paper they said, well, maximum response, we can just control that, and the shape is just going to be determined by the Rmax, which we can fix, and just, you know, look at the inflection point. But you can kind of see here that really some of the binding is really, you know, far off. So how accurate is this? So do you have the right range in the amount of data that you've got? Sometimes it's very simple to kind of be fooled just because of natural variation in the data. Okay, here's some data, and if we take a log view, we can kind of see we get some nice plateaus here, and in the middle, then we've got the binding. So what happens if we then just take a limited range? It's going to take all the same data here, limited range, and a limited number of points. Okay, variation. We can fit a curve. That's very nice, but essentially we haven't gone through to the plateau at the top, and that the KD we're getting is pretty much just above the total concentration of the protein that we've put in. So that's always a warning sign. If your KD is similar to the top concentration, you haven't gone far enough. Alternatively, then, if you take too short a range, we just kind of go into, well, we haven't gone far enough. We're just going to take this data range here. Again, wow, we can fit that. If we look at the log view, we can see we've got reduced amplitude, and actually the KD is now much lower than it should be. So often people will kind of want to try and get nice curves by just doing normalization. You kind of say, okay, I know I've finished my assay. This must be saturation. So let's just normalize the data, because I can just do a normalization by the fraction bound and fit that. But of course, the problem is that if you haven't gone far enough, then you're completely off the scale completely. It's always best to fit raw data. Time is another thing that again is something that a lot of people don't kind of think about. You know, just titrate out into your microtiter plate, stick it in the plate reader, press go, it reads, you get a thing. Here's a bit of an extreme example where the assay was then kind of left for quite a long time. And I think this was even overnight. And you can kind of see that what's happening is that we're not only getting a larger amplitude, but also the KD is getting tighter and tighter. So a good thing to do is that in those kind of assays is, you know, set up your titration, leave it for a bit, leave it for half an hour, a couple of hours. In kinetics, it's also then, you know, take the long view as well, is kind of say, you know, have I reached, you know, saturation to be able to fit a good exponential here? Has the endpoint been reached? So if we, again, just take the initial kind of couple of seconds, oh, we can kind of fit, oh, we can fit a data here. But actually, you know, if we then plot that out, we've really got the completely wrong amplitude. And this can be an issue then in things like Biophysical and octet, where you really, in very slow kinetics, where it's coming off very slow, you can't really get to the end. And we'll see later an example of getting things wrong that way. So the rule of thumb in this case is like 10 to 20 fold the T1/2. Enzyme kinetics, it's the other way around. If you're wanting to then do mechanical kinetics, you're just assuming that there's no product and that you've got the enzyme is much lower concentration substrate. So the substrate is basically, the concentration is free. And you're only looking at the initial kind of rates here, because you then hardly any product has been made. Okay, so do you have appropriate concentrations? It depends upon how you kind of fit things. So in this case, if we're doing, say, fluorescence in FP and MST, usually the ligand is at a much lower concentration than the KD. So that would then, when we titrate in a protein or something else, that doesn't really change as the concentration of the complex is really small. In cases like that, where if you get the binding becomes really super tight, and that it's essentially the half the amount of the total amount of, say, protein or ligand you put in, then it means essentially you got into stoichiometric titration, and that you need to then reconfigure. ITC, it's the other way around. You want a stoichiometric titration so that the amount of, say, protein in a cell is way above the KD, 10 to 20-fold, and so that P titrates, the protein titrates as you add more ligand in. The issue there, of course, is that you need to know roughly where your KD is. And as always, have you measured your concentrations correctly? Okay, so you've got everything is good, you've got everything in the correct units, you've got all these assumptions are good. Let's get on to the data model. So the questions in this is, well, is my data good enough? Do I have the right equation? How good is the fit? Does this correlate with what you think the physical model actually is? So here's a data model, just simple binding, and we're going to assume then that the change in fluorescence is directly proportional to the concentration of complex. Don't worry about the details here, but you're working through the KD. You're making the assumption that the change in the fluorescence is directly proportional to the concentration of complex, and that when you get to the maximum fluorescence, that's going to be equal to the total amount of protein that you've actually put in. So you substitute that in, give it this kind of form here, which is in a form of y equals some kind of function against x, where x is the concentration of whatever you're titrating in against the parameters. So in general, we do non-least squares fitting, where you have a dependent variable, so that's your y or your signal, and then you've got an independent variable. You've measured that protein concentration, you've got the parameters, and we assume that you've got accurate concentrations. It's uniformly distributed across everything, the concentration range, and you assume that the dependent variable here, you've got the variability is really Gaussian, it's random. And so what the fitting programs will do is they'll try and minimize the difference between the fitted parameter and the actual data here. And so it does that by taking the sum of the squares of the residuals, and then goes around and then changes the parameters, goes around and sees, well, what's the then sum of the squares then, changes the parameters, goes round and round like that to try and minimize things. And here would be just an example where just simple binding. Here's kind of one round. You've got some, you need to kind of initialize with initial parameters, so it kind of sort of fits. You kind of know where you are. 2 rounds, getting better, and then 5 rounds, 10 rounds, it's getting even better, and then 5,000 rounds, it fits completely. So what it's trying to do, if you've got just one parameter kind of changing, then it's looking for this kind of minimum point here. Of course, if you've got like 2 parameters changing, then you've got a surface, 2-dimensional surface, and then, but a lot of the times you'll have multi-parameters, and so you're looking at some kind of large, higher dimensional error space that it's trying to go. Of course, the problem, the more parameters, then sometimes what can happen, so initially then you're going to go down to this minimum, but if you've got more parameters, sometimes you can get then fitted into this kind of local minimum and not be able to get out. So here's an example of a data model and how we'd kind of start to fit things. So, this is one from the fluorescence polarization. And in this case, then we're titrating in, but we're also at a concentration where we're going to titrate the labeled ligand in as well. So that's going to change. So we need to take account of that. So everything is changing. And so we end up with this kind of quadratic equation here. So if we kind of fit in with this program called Kaleidograph, we've got things in data columns, and certainly I like to use this. This has kind of got nice transformation tools. It's very, very easy to use. You can certainly plot the data. There's lots of saved models here you can write yourself. And so, I mean, one of the issues here is, of course, is that you have to then write in your own equation here, and it's in terms of m. So this then changes into this when you put it in, and you also then need to put in some estimates of where you think, idea of what the KD is and what maybe the top kind of saturation value is. Of course, if you put the bad estimates in, it's just pretty wrong. It will fit, and you'll get a correlation of 1, but it's just terrible. So then what you get out of this in terms of you get the data values out, you get an error on the fitted values, you get a chi-squared, and also a correlation coefficient. So what are those? We'll come to those in a minute or two. First of all, we'll kind of show you probably something that you're probably more likely to use, which is Prism. And again, it's kind of got things in data columns, and we can do non-linear regression here. So we'll do. There's lots of then standard equations, and you can make your own equations here. The nice thing about this is that when you write your own equations, you can use words. So it's pretty easy to kind of say, right, here's this same equation here we used earlier, but we can just put this into words and say, well, this is y is equal to bottom. That's the bottom of the fluorescence or the polarization, and top left bottom, the square root, and B and C, and you can define what those are. And in this case, it gives you automatic estimates, so you can put into the formulation of the equation where you kind of say, well, okay, I'm going to say the bottom is y min, top's going to be y max, KD somewhere in the middle. And you can check the values by also kind of look to see if they're reasonable, just by clicking at the tab here. And then it'll give you a fit similar to Kaleidograph, where it gives you the fitted values here. It will give you what the standard error is on these ones. And then it'll give you confidence limits. So in this case, it's KD here will be between, even though it's 1.9, it says, well, confidence at 95% is 1.4 to 2.34. The goodness of fit here is it's going to be R squared rather than R. So what's chi-squared? Well, essentially then chi-squared is really how well your data is fitting to the how well the fitted parameters actually match what your data is as well. Sometimes you have this you can put this weighting function in here, but usually without weighting, I mean, this thing is 1, and you only really need to weight if, say, you've got a difference variation in your error as say the concentration increases, or so you don't put that in just to fit better bad data. And in general, the lower the chi-squared, then the better, because that means that then your fitted parameters are fitting the data much better. And then the correlation coefficient is then really a ratio of the chi-squared then divided by then these values here, and that is really the kind of a null hypothesis, which says that say there's nothing happening, you're not getting any binding at all. If we kind of look at what the difference of all your data values are between an average value of y, how does that then compare to the chi-squared? And so obviously, if you get something closer to 1, then that's going to give a perfect condition. Things then below 0.7, then you have to use with caution. So how good is the fit? So we're looking at really, you can look at, say, how big these errors are compared to the values. How well so that's kind of telling you how well this parameter is actually constrained. If you get high errors, is this, you need better data? Is the model not correct? Hopefully you're going to get a low chi-squared and a high R. And the big question is, are these physically relevant? Do they have significant values, or can you actually leave them out and have a more simple model? Would that be actually better? And a big thing, as we'll see later, the errors here are actually fits to the data. So these are mathematical calculations. It's not telling you what the variation in how well your data is. It's not the accuracy or the precision. So not the variation in your binding constant. In this case here, I'm saying, yeah, it's probably M4 here. That's not, the overall error. That's just how well things fit. So the good idea is then to visually inspect everything. Often you can kind of tell how well things are fitting just by looking at it. And again, in this case, take a log view. Similarly with kinetic data, look at say other things. You can also do residuals, and these can then be plotted from in Kaleidograph. Then just in the fit curve section, you can actually output the residuals, and in Prism, then in the diagnostics, you can automatically then plot them. So then this gives you an idea of, well, how well is my model actually fitting? In this case here, we've got nucleotide binding kinetics, and a single kind of exponential isn't really fitting. There seems to be some variation here. If we put a bit of double in here, then again, the double's not really fitting as well. So then we've got a bit, maybe another function, bit of drift here in the signal, and that fits better. If you've got a systematic deviation in your data, then you've got to really think about, well, do we change the physical model of what's actually happening? One thing you don't want to do is you don't want to have bad data and then think, I'm just going to put more parameters in to fit it. Here we go. Here's an example where someone came with, with 5 data points. Yeah, let's fit it with this model here where we've got binding, and then we've got this non-specific binding term. Oh, it fits really well. Well, how would you improve that? Or you get really big kind of, pinches and gaps. And this is a really kind of astounding data that was published recently. And this is backward data. And so in the fin, the thick data here is the data. I think the fin is the fits. I think even at the back there, you can probably see that it's not really fitting at all. But even so, it was fitted, and they kind of then quoted everything to 4 or 5 decimal places, and they even said that this R squared was 0.93, which makes me think, really? So, it's obviously not a one-to-one fitting. Of course, bad data, sometimes you get outliers. So how do we kind of judge, what is bad data, or, are we kind of cheating? So sometimes, you might get stuff here where, it's really, really outside everything. Sometimes that's necessary. In ITC, what we do is we always do an initial little injection here. And that's because you have a pipette in the solution and it's sitting there while it's spinning, and sometimes you then get exchange between what's in the cell and what's in the syringe. And so we know that we're going to discard that point, so we can get rid of it. We know the reasons for that, and then when we get rid of it, it's like, yep, that's fine. We can fit everything. So over time, you kind of go, well, there's these points here at the top. Yeah, I don't like these. Let's get rid of those. But maybe you need kind of a bigger data range, and actually see. In this case, yep, go on. There's another binding step happening here. So Be careful about that. So, is this kind of deviation here kind of seen, only in kind of 1% of the data? Is it just random? Is there something else happening? Is this do you need better buffer conditions? Is there something wrong with the data or actually the data model? Okay, so now we'll come on to kind of we've got this quality of fit, everything's good. Yeah. Sorry, can you just you won't be able to be heard, so yeah.
Yeah, so in the past I was working with a protein which had a two-step binding process, but we could not go high enough with protein concentration to get the second KD because this was millimolar range. So in this case, is it okay if I just remove these data points and try to fit the first KD?
Yeah.
So what's your thoughts on that?
Okay, yeah, in conditions where, say, one of the components, we often come across this, and where one of the components you just can't get it up to the concentration because it just will aggregate. The thing to do then is just either change technique or swap everything around where you're then titrating in the other component, as long as you can kind of get enough of that. So, I mean, this would be a case in say ITC, where sometimes you can't get up, if you've got something in the syringe, you can't get it up to that concentration, you can swap things around, or in Biocor, where one of the components is then got to be on the surface and the other one's going to be flowing across. So you can swap that around and have the one that's going to aggregate kind of on the surface. So, right. Okay, so what things are not fitting? Do you have nonsense, parameters, really, really bad? So what can you do? Well, sometimes in PRISM, you can do things like constrain. So let's say mathematically things are going off. Often if you think about those peaks, you're getting into some remote, higher minimum point. Sometimes you could constrain a point. So, definitely a KD is going to be greater than 0. We know that. I mean, this is an example here of doing also, you can do shared values as well. And this is a case like as you kind of had where titrating in the same protein then into a protein that was either non-phosphorylated or phosphorylated. And here I couldn't get up to the right range here in the blue, because essentially that when you got up to that concentration, the protein started to aggregate. This is an FP, fluorescence polarization assay. And I could see the total intensity of the light was increasing above these concentrations. That meant that the protein was aggregating. So what can you do? Well, in this case, then I could say, well, I know it's going to be the same protein. And I know that when it binds on the other protein, I should reach the same maximum here. So obviously it's not ideal, but what we could do, but you could get then a relative KD out. But also it was important that it was explicit in the methodology that I said I did this, and not kind of say, right, let's just fit that to get better data. Often then we can, instead of doing fitting individually, we can do global fitting. So we can share these parameters. Sometimes that's needed if, say, you're doing kinetic data with SPR. You might have experimental, variation. But it's important to kind of think about, well, how do the global fits compare to the individual fits? So here with Biocor, then we can fit this either 2 ways. We can fit this data either with the on and the off rates to get the KD, or we can look at the equilibrium position here, and this is equilibrium response, and this is the residuals. So if we fit globally, we get these things in the blue, and then go for the on and off rates, and then in red here is the individual fits, and then this is an average here. So the question would be, okay, well, are these outliers or not? So it could be that, well, what's actually happening in this case? In this case here, the outliers, it could be that these are for very low concentrations. So we've got not very, very large amplitudes, and it could be that it's just not fitting very well in those kind of positions. In that case, then, we might be justified in saying, well, actually, these are these outliers are actually just bad data, and we can discard them. Or it could be that this is the natural variation, you need to include it, and it's just, well, this is just a variation in terms of, and also you can kind of then compare the KD that you get from the association rates into association rates against the KD you get at equilibrium and compare. And here's an example where global fitting can get to error. In this case, again, this is a reproduction of a published method, and this is again with SPR, and the global fitting gave a very, very low K off. It fitted very nicely. That was all very nice. So give an estimated KD of 30 nanomolar. That looked very, very tight. But if you fit it then to the individual association rates to then get observed rate constants, you could get the KD of 1.8 micromolar, and from ITC is 2.2. So as we've seen in some examples before, then just because the program fits and the computer says yes, and you get how many decimal points of kind of fitting, doesn't necessarily mean that you've actually got the right answer. So always be cautious. Okay, so everything is all nice. You just repeat, repeat, repeat. So let's think about biological versus technical replicates. Okay, so would be both a biological and a technical replicate of the fluorescence polarization data, and you can see that it's all pretty much over different days, different samples. It's all pretty good. One question we had is, well, you've done all this, you've repeated it, blah blah blah, different times, and shouldn't you just then average everything together and then take the average of that? And that would be the question of no, because you're then discounting the fact that you're maybe certainly getting different amplitudes in different days. So don't ever do that. In terms of biological and technical replicates, again we don't tend to do things like high throughput screening. But here's an example of this assay again, high throughput screening fluorescence polarization. So these were different preps, batch 1, batch 2, batch 3, and these were different than technical replicates. You kind of see as I purified the protein better, and then the person doing the high throughput screening formulated the assay, it got better and better. So in both these programs, you can do kinetics, in both these programs, you can do statistics. So you can take all your values, and then you can just fit and get statistics for everything. And similarly in PRISM as well, there's lots of different ways of actually then looking at your confidence limits or your standard deviations, et cetera, and you plot those all out. But of course, the big question is, what does it all mean in the end? In terms of binding constants, well, we're looking at then energy differences. So, in terms of a log view, how significantly different is if its binding constant is twice or 3 times. Okay, so sometimes you have to think about, well, when you're constraining the fit, you assume everything is the same, and you've got your distribution is not going to affect the fitting, but sometimes within fits, then some points are more important than others, and this is why say transforming in the old ways to transform into linear plots like the Lin-Weaver-Burke and stuff that you did in your undergraduates potentially. Don't do that, because essentially you're dividing the concentration by, say, a signal change. So, and then that means that some of the points are really more significant and will really determine the data fitting. So always fit raw data if you can. And there's various other programs to kind of resample your data, and like bootstrap and jackknifing, to see how the points determine the fit. Propagation of errors, if you need to kind of add things together, then it's simply just a square root of the sum of the squares of the deviation or standard mean. But if you multiply together, then you need to then do this formula here, where it is the the square root of the sum of the square of the ratio of your deviation against what your parameter is. Obviously good, you've got binding due to kind of different techniques, compare, say, in this case, like comparing ITC against fluorescence, and of course, and repeat. And then hopefully you've got something that will be meaningful. And in the end, then the two takeaway messages are try and keep things simple if you can. Don't invoke more complex things if you just to fit your data better. But on the other hand, then maybe things aren't so simple, and if you're getting again and again, you're getting deviations that maybe are going away from what the model should be. Kind of lift up that carpet, and what Sidney Brenner has kind of termed Occam's broom. Take the dust out and actually see, is something different going on here? Am I kind of biased against this kind of deviation because something new is happening? Okay, so that's for today. Any questions anybody's got?
So the question, like very often people would want to do ITC and SPR or something, and then sometimes they're within like a log-fold kind of difference, and then people just give that range. Any advice there?
In terms of what? In terms of the log?
Say like, oh, this is between 0.1 and 1. The KD, we think
in terms
because one gives 0.1 and the other one gives towards 1. So we say, oh, that's the range between the one and the other.
Yeah, I mean,
advice there.
Advice in terms. Well, I mean, if say some things are say 2 or 3-fold different, I mean that's not really that much significant in terms of binding constant, because the binding constant is then the energy is a log form or ln form. That's not much difference in terms of the amount of energy that's actually happening. So that could just be whatever the assumptions you've made in that technique, or maybe your concentrations are not exact as you expect. So you can kind of get that variation. So I mean, I think in terms of you just quote things and say by ITC it was this, and when we repeated it by this we got that. So this correlates with this by that, because you assume if say something was like 2.5 and it was 5 by ITC or something, then that's kind of your variation. I mean, you could say, and sometimes then you do get confidence limits. If we kind of go back and say in the prism, so it will say it will fit something, and then it will say, and again I think ITC as well, I think we'll give confidence limits as well, where it will say, this is a KD, but the confidence limit of 95% is it's going to be between 2.4 and 6 or something. So again, you can, in those kind of circumstances, you could be confident that when you get a slightly different result by another technique, that's still within a good confidence limit.
And if it's not?
If it's not, go and repeat.
So I just had a question about so the replicates.
Yeah.
On separate days, for example, which is something that happens all the time, and you do see differences.
Yeah.
What is an appropriate way of dealing with that? Would that then be a case of look at the graphs separately,
yeah,
assess them individually, and then normalize internally and then combine? Would that be?
No, that's a no-no. So that's a no-no. I mean, if you say, if I kind of go back to, so this is a case in point. Okay, this is done. So within each of these graphs, then there's a triplicate, right? So we've measured the signal 3 times, and then it's done on different days and different preps. So you might get natural variation, and that could be that your protein isn't kind of is as stable as you think. Maybe if you're say taking it from -80, and then it's you're sure that it's the same, but maybe you've prepped it, you've done that once, done it the next day, and so I would say an important thing about that would be to ensure initially to make sure how good is your protein or your other kind of components. Does it kind of go off? So that you're getting this variation over kind of days. So I think and we've got lots of tools to be able to look at that. How stable is your protein in the buffer system that you've got? Is there something better to do? So yeah, so always, again, if you're here in this case, then you're fitting the data individually, and then you're getting KDs out of that. And then from those KDs and your statistics on that to then give you what your standard error is then the question is then, what would you present in a paper? I mean, you can't present all 5. So, I mean, also you could put them into the supplementary, but often it would be a case of going, not necessarily the best looking data, but the data that kind of best represents, your average, whatever your average value, and that's the way I would kind of look at it.